Hello, and welcome to our very important discussion today on cholesterol metabolism, particularly LDL receptor metabolism, low density lipoprotein receptor metabolism. What is the relationship between the liver cell hepatocytes and the metabolism of cholesterol esters, triglycerides, and the packaging and the preparations of these lipoproteins and delivering them to the fat tissue, the adipose tissue, as well as the blood vessels, and delivering the fat tissue, and delivering the cholesterol, which is a form of energy, to the skeletal muscles. And then we're going to look in depth. We're going to look in depth on all the processes that unfold inside the cell. How does the endoplasmic reticulum participate in the making of the LDL receptors? And how does the cell regulate the synthesis of the LDL receptors on the extracellular plasma membrane? And how um, your cell is able to endocytose and bring internally the cholesterol that accumulates in the bloodstream. So let's take everything step by step. So what do we see here? So here we see a liver cell. I'm pointing to a liver cell, and these are the hepatocytes. Yellow represents triglycerides. We will talk about what those are. Triglycerides. Triglycerides are in yellow. In red, we have cholesterol esters. In red, you have cholesterol esters. So we identified the liver cell, and the liver cell bursts out and provides us with a complex known as VLDL. We will get into that, but for now, we're identifying our players. So VLDL is the very first component that comes out of the liver cell. And it's a mixture of yellow and red, with more yellow than there is red. That is very important. There's more yellow circles, more yellow balloons, more yellow blebs, more yellow bubbles than there are red bubbles. So red stands for cholesterol ester, while yellow stands for triglycerides. And that is the reason why we call them very low density lipoproteins, because they have more ratio, more proportion of yellow, more proportion of triglycerides than do they ever have red. Later on, that's going to reverse. So we're going to see a complex known as LDL, low density lipoprotein. That one is now going to have more red cholesterol esters than yellow triglycerides. That's the bad cholesterol. Bad cholesterol has more cholesterol esters than it does triglycerides. And we will get into that. So, what happens is when you take in a meal, when you eat a big beef burger that has a lot of lipids, and the lipids are obviously going to be metabolized. You know, from our metabolism, we learn that we metabolize sugars through the glycolytic pathway to get ATP. We metabolize proteins through protein catabolism pathways to get ATP. We metabolize nucleic acids to get ATP, and we metabolize lipids to get ATP. But now we're going to look more in depth in this process of metabolizing the lipids. So, what happens is the liver cell has these green receptors. You see, I'm pointing to these green receptors. These green receptors, they are able to bring in, swallow, gallop, endocytose, bring in LDL, bring in low density lipoprotein. That's why they call them LDL receptors. They call them LDL receptors because they receive the LDL and bring it in from the outside. That is very important. From the outside is the blood, inside is the cell. So they bring in fat from the blood to the cell. So you expect there is a lot of LDL receptors after a meal. When you eating the beef burger and there is in your blood a lot of lipids. Obviously, we need to internalize and bring in those lipids inside the cell. So your cell is going to be making on demand a lot of LDL receptors to start bringing the lipids from the outside, the blood, into the inside, the core of the cell. So once this low density lipoprotein has been brought inside the cell, now the liver has the enzymes to repackage them. Are there cholesterol esters? Put them on one side. Are there triglycerides? Put them on the other side. And when you're burning energy and you need more energy, there's a lot of energy expenditure. So when you need energy, your liver, even though your liver received LDL, it does not mean when you need fat, it's going to give you LDL. Even though it took it in as LDL, it's not going to give it out as LDL. It will give it out as VLDL, very low density lipoprotein. That is very important for you to understand that when my liver, who is the storage site of lipids, it's obviously also the storage site of sugars and proteins, but for now, it is we're doing lipid metabolism, so obviously we're going to store a lot of fat. So when your body needs to burn down fat, your liver is going to bring out this fat in what form? HDL, LDL, or VLDL? That is going to be VLDL. Now, VLDL, why is it very low density lipoprotein? Because it has more yellow than red, because it has more yellow triglycerides than it does have red cholesterol esters. Why is the LDL called LDL, low density lipoprotein? Because it has more red. It has more red inside it than does it have yellow. Obviously, there's both of them, yellow triglycerides and red cholesterol esters, but the ratios are different. In VLDL, there is more triglycerides than there are cholesterol esters, but in LDL, there is more cholesterol esters than there are triglycerides. So what are we now? We're now on the first step, and that is sending out VLDL. Now, this VLDL is going to come out in a lipoprotein. So it's like a submarine. The submarine from the outside can swim in the ocean, but in the core of the submarine, you have the people who cannot dive in the ocean. So obviously, the blood is polar. The blood is polar. The blood does not dissolve the nonpolar lipids. And obviously, we know the lipids are nonpolar. So we cannot dissolve nonpolar into the polar blood. So we have to, when we do drugs, for example, in pharmaceutics, what happens is your liver is going to convert the non polar drug into polar substance. That's known as activation. That's known as metabolism of the drug. But this is not a drug, that's a lipid. So what it's going to do now is it's going to put it in a package known as this VLDL. So it's a very low density lipoprotein. It's a hybrid. It's a two-in-one lipoprotein. Lipoprotein. Lipid is one thing, protein is another. You put them together, you get a mixture, a hybrid, lipoprotein. Obviously, where is the protein? Is it in the core or is it on the outside? Obviously, the protein is on the outside because the protein has the amino acids, and the amino acids have charged R groups, and these are the ones who are going to be ionized in the blood, and that's what's going to solubilize the VLDL in the blood. But in the core, you're going to have your triglycerides and your cholesterol esters. Then we're gonna dive in the blood, and obviously we see these horns, these triangles, this red APOE. What is this B100? What is APOC? These are all receptors, recognition receptors. If you look here, this LDL receptor, the LDL receptor was able to recognize LDL, bind it, and internalize it, bring it inside the liver cell by grabbing these horns, by grabbing these triangles, these ligands known as the B100, the APOC. Then we're gonna move now. So now we have step one, the very low density lipoprotein just came out the liver and it's gonna go into the, you see this blood vessel? This in the middle here, that's my blood vessel. In the middle here, that's my blood vessel. Once the very low density lipoprotein is inside the blood vessel, it has one of two options. It can either go here in this structure, and that is the adipose tissue. This is the adipose tissue. We can store the fat, the VLDL, in the adipose tissue, or we can send it into the skeletal muscle. You see, that's a skeletal muscle. So the VLDL came out the liver. It's gonna go, the first thing it's gonna do right after coming out the liver is gonna go into the blood vessels. Inside the blood vessels, there are prepared enzymes. These enzymes known as lipases. ACE is an enzyme. These are lipases who will hydrolyze the lipids. So these lipases, these are known as endothelial because they are inside the endothelium, the blood vessel. These are known as endothelial lipases. They will solubilize and they will hydrolyze the VLDL and remove the cholesterol esters and remove the triglyceride and start giving them to the skeletal muscle if you are running, so we can give you energy so you can burn down the ATP and maintain your running pace. But if you're not burning this lipid, what's going to happen is it's going to go right into the adipose tissue and get stored there. And the very last option is we stored too much fat in the adipose tissue and we gave a lot of fat into the muscular tissue. Now there is some remnant of this VLDL that has been hydrolyzed with the lipases. So now you have something known as IDL, not yet LDL. LDL is low density lipoprotein. Now you have a leftover that came out of the blood vessel known as IDL. IDL is known as intermediate density lipoprotein. Now, intermediate density lipoprotein has a little more red than yellow now. Now we tip the balance. In very low density lipoprotein, there was more yellow than red. But in IDL, there is more red than yellow. In LDL is more red than ever will be yellow. But IDL is a transition phase, intermediate phase between VLDL and LDL. So the IDL can be immediately, there's two pathways now. There is pathway A, and there is pathway B. Pathway A, you're going to have IDL itself going and being brought inside the cell because the IDL does have this green horn, this green recognition factor known as the B100. It'll be taken inside the liver cell as if it is LDL, even though it is IDL. But the LDL receptor will not care because the IDL has that green part to internalize it. That's how IDL goes in. That's the first path, A. The second path, B, we will now convert the IDL into LDL. So obviously you see LDL is all red. There's no yellow. And then the LDL is going to be brought inside the liver through receptor mediated endocytosis of the LDL. That is receptor mediated clearance of LDL. And then some LDL will remain in the blood. It will be cleared in other ways. So this is a very important overview of what happens in our cholesterol. It's going to be first brought out of the liver in the form of VLDL. Then it's going to be distributed based on the demand to the skeletal muscle, and some of it will be stored in the fat adipose tissue, but the remaining will come out as IDL with obviously more cholesterol because your body utilizes more triglycerides than cholesterol. But if you have too much cholesterol anyways, now we have no way but then to convert it into LDL that has a low density because it has more cholesterol than triglycerides, and it'll be stored inside the liver. So now let us now look into the cell. Let us now examine what happens once this cholesterol has been brought inside the cell. What are the ramifications? What are the effects? What are the effects? So now let us examine what are the effects. Let's move on to this side. So now let's examine what happens inside the cell. So let's first recognize our players. We've already mentioned that this green part here, that is the LDL receptor. That's what's going to internalize and bring inwardly. That's what's going to internalize and bring to the inside. That's what's going to internalize and bring into the inside the LDL. So if there's a lot of LDL, low density lipoprotein, is going to be brought in with this green receptors. And how will it be brought in? If we have LDL receptor, is going to be brought in through clathrin coated pits. So this red clathrin protein is going to be enabling this vesicle endocytosis. So what happens is that's your LDL. You see LDL? LDL has a lot of red bubbles. You see the red bubbles, you see the red cholesterol, and you see the green horn, ApoB 100. That's gonna bind our V-shaped. You see, you're gonna find the LDL receptor binding the V-shaped um, ApoB LDL that's carrying a lot of the cholesterol. Once it binds, it's going to be brought in. You see, that is your coated pit. The coated pit is going to be a bubble that has the LDL, and you see the red cholesterol is in the core. Then the red cholesterol is going to be sent into the endosome, and the endosome is going to send this package that has a lot of cholesterol inside it into the lysosome, and the lysosome is going to break apart this vesicle and remove the outer coating, and then liberate cholesterol. You see, now that's how we got cholesterol. So that is one pathway to get cholesterol, and that is the endocytosis mediated clathrin coated pit. The clathrin coated pit is going to bring in the cholesterol and deposit it in the cell. So that means if the cell has already too much cholesterol, the cell will deactivate the synthesis and the making of LDL receptors so that no more LDL is needed inside the cell. Imagine there is already too much cholesterol inside the cell, and the cell keeps on making more LDL receptor. Receptor means bringing in. So if we make more LDL receptors, we're bringing in more cholesterol, more cholesterol, more cholesterol, more cholesterol, and we already have a lot of cholesterol. The cells are going to be laden with so much cholesterol, there's going to be stress. So one of the ways the cell regulates cholesterol metabolism is by increasing LDL receptors when it needs cholesterol and inhibiting the synthesis of LDL receptor when it no longer needs cholesterol. The second way is that the cell can endogenously, from the inside, endogenously make cholesterol through this HMG-CoA reductase. HMG-CoA reductase, very, very, very important enzyme. HMG-CoA reductase. So this HMG-CoA reductase will directly synthesize cholesterol inside the cell. So if we need cholesterol inside the cell, we turn on, we activate HMG CoA reductase, and then we get cholesterol. If we have already too much cholesterol, then we inhibit HMG CoA reductase, and that will stop the making of cholesterol because we already have enough. And cholesterol is going to be ultimately stored, whether it was brought in by clathrin coated pit from the outside through the LDL receptor, or it was endogenously made in by the HMG CoA reductase. Is going to be stored inside the cell. And if we have, now let's look at another way that the cell regulates cholesterol. So if we have too much, if you have oversupply of cholesterol, what happens is the cell will be stimulated to store this cholesterol. So cholesterol is not going to be just dispersed all over the place. It's going to be packaged and stored inside the cell. Also, the cell is going to inhibit, we talked about that. It's going to inhibit the synthesis of LDL receptor. So the cell, if we don't want cholesterol, we would inhibit DNA transcription, because LDL receptor in the end of the day is a protein, and you can never get a protein unless you transcribe the DNA. And when you transcribe the DNA, you get RNA. And when you translate the RNA in the ribosome, you get the protein of interest. So if we want to stop bringing in cholesterol, we need to stop the transcription of the genes that will give us the mRNA that once translated will give us the protein LDL receptor. So we inhibit the synthesis of LDL receptors by oversupply of cholesterol would lead to the inhibition of the DNA transcription that leads to the RNA, the RNA transcript of LDL receptors that would ultimately be translated in this is the endoplasmic reticulum, ER. So normally when we don't have cholesterol, the cell was going to turn on the DNA transcription of RNA, and RNA will be translated to give us LDL receptor. So the LDL receptor is going to be synthesized in these ribosomes. These purple bodies are the ribosomes on the endoplasmic reticulum, and then you see the green is made. The green LDL receptor is made in the endoplasmic reticulum, and then you see really well here that this you see that these vesicles containing the LDL receptor are going to be taken into the Golgi body, and the Golgi body is going to make the exocytosis of, you see, this is exocytosis, bringing out the LDL receptors as we see here and plucking them on the plasma membrane of the cell. So that shows you now we regulate uh, endocytosis, bringing in of cholesterol by transcription of the DNA. So when the opposite is there, when we already have too much, we don't need cholesterol, we inhibit the DNA transcription of RNA, and we no longer be able to make proteins that are LDL receptors, because we inhibited the DNA transcription and we inhibited the RNA translation that will ultimately give us the LDL receptors. Lastly, I would like to introduce this PCSK9. What is PCSK9? PCSK9 is protein convertase subtilizing kexin 9. Protein convertase subtilizing kexin 9. What is protein convertase subtilizing kexin 9? This protein convertase subtilizing kexin 9, PCSK9, is an enzyme that is responsible for destroying and degrading the LDL receptor. So usually on the exam, they don't tell you about how does it influence the cholesterol metabolism. All that they need to tell you is protein convertase subtilizing kexin 9 is an enzyme that is responsible for the destruction of the LDL receptor. So you know, since PCSK9 destroys the LDL receptor, that means if there's no LDL receptor, how am I going to be able to bring in cholesterol inside the cell? So that's a way for me to regulate the bringing in of cholesterol. So if I have no cholesterol inside the cell, I need LDL receptors. So obviously, PCSK9 has to be inactivated. Because if PCSK9 is activated, it's going to destroy the LDL receptors. And I'm not going to be able to bring in cholesterol without the LDL receptors. So I'm going to turn off my PCSK9 in order to bring in cholesterol, LDL. But if I have too much cholesterol, I have too much LDL inside the cell, then if I leave it on, if I leave PCSK9, if I leave it off, LDL is going to be plucked on my membrane, plasma membrane. We're going to bring in a lot of cholesterol. We don't want that. We don't want the stress. So we're going to turn it on so we can destroy for us. You see, it has an X on this LDL receptor. So it's this is a very important step into the understanding of how the LDL receptors are recycled and used as a way to monitor the metabolism of cholesterol. Thank you very much, and this is I'll see you in the next video.